Grade 12, so we're going to look at DNA, the code of life, as the first topic for the year. Now, DNA is a very important part of our genetic makeup, of everything that we do, because DNA ultimately tells a lot of characteristics about us, from our genetic illnesses to whether we will have hormones in abundance or hormones that are not in abundance, whether you'll have cancer one day, whether diabetes, high blood pressure, hypertension, which is known as hypertension, runs in your family, and all of those types of things, hair follicles, eye color. All of those things are coded for by, or not are coded for by genes, but DNA is the starting point. But I'm not going to speak too much, we'll get into that a bit later. But firstly, let's look at DNA. Now, we're going to first look at some revision from grade 10. Now, the revision from grade 10 will consist of the structure of the nucleus. And the reason why specifically the structure of the nucleus is looked at is because the nucleus houses, in layman's terms, houses DNA. It is where DNA is found. And that is why we have to look at the structure of the nucleus, because the nucleus ultimately is the starting point of DNA. Now, if we look at the structure, we can see that we have a nucleopore, a double nuclear membrane, nucleoplasm, chromatin network, rough, endoplasmic reticulum, and ribosomes. But the important structures that we want to take note of here is your ribosomes, your chromatin network, your nucleoplasm, and your nucleopores. And these are all going to play various roles in later topics. For example, chromatin network will play a topic in meiosis, which is the opposite of mitosis. Mitosis is the division of somatic cells, and meiosis is the division of gametes or sex cells. And ribosomes we know is the site of something called protein synthesis. And we're going to look at that as well to come in this topic a little bit later. Right? And the nucleoplasm is the cytoplasm like fluid. It is similar to the cytoplasm, and it is the fluid cavity in which the organelle inside the nucleus, your chromatin network, your nucleolus, that black over there is known as the nucleolus. Right? It is the fluid membrane in which all of this organelle is found, and your nucleopore will also play a role in protein synthesis, where it makes a pathway. It makes a pathway, an escapable pathway for something called mRNA, which we will look at later in this video. Furthermore, now we're going to look at the functions of the nucleus. Now, the function of the nucleus: chromosomes inside the nucleus store genetic material of the cell. Why did I say this? We know that a chromosome looks something like that. Whether unreplicated or replicated, okay, not whether unreplicated or replicated, but now because of the changes in the curriculum, it's just a chromosome or chromatid, depending on which topic you're doing. But chromosomes inside the nucleus store genetic material. Now we know that DNA is housed within the nucleus, but it is a grade 10 understanding. The grade 12 understanding comes in here now, that it is chromosomes. And the reason why that is important, I'm going to show you in a later video, and then I'll link it back here, is that from the chromatin network, it will thicken, it will unwind, it will condense, and then it will become a chromosome eventually. And one segment, a small part of this chromosome, is DNA. And a small segment of DNA is known as a gene, and we're going to look at all of those stuff a little bit later. But that is one of the main functions of the nucleus. Another function is the nucleus regulates both structure and metabolism of the cell. The nucleus is the organelle that makes sure that the metabolic activities of the cell, like protein synthesis, for example, is being done, or osmoregulation is done within the cell. It also maintains the structural integrity of our cells, that our cells are not damaged um, by pathogens and antibodies and all of those types of things which we looked at last year. But simple enough, the nucleus regulates both structure and metabolism of the cell. And that is all the revision that you need to know from grade 10. There's no revision from grade 11. Now, what we are actually starting with in grade 12 is something called a nucleic acid. Right? Now, a nucleic acid is an organic molecule. Now, for those of us who do organic chemistry, we will know that it's an organic molecule because it contains carbon atoms. It has carbon atoms, and that is why we call it an organic molecule. But that you don't need to know. That's more for interest sake. And then also we want to know that it controls protein synthesis. And protein synthesis simply just means that the body is manufacturing, or living cells are manufacturing proteins. So it just means that proteins are being made. Proteins are made, and that is simply what protein synthesis is. Anytime you see the word synthesis as a suffix, it means that something is usually being made. In photosynthesis, we saw that something was being made in the presence of light. Here we can see that proteins are being made in living cells. And we have two types of nucleic acids, and this is where we actually start. You have your DNA and you have your RNA. Now, from grade 10 and grade 11, we learned small facts about DNA and RNA. We learned that it's made up of something called CHON-Per, where you have carbon, hydrogen, oxygen, nitrogen, and phosphorus. It is made up of those elements. Various other elements, but those are the main elements that is looked at in medicine and in chemistry. Those are ultimately the 5 elements that make up your DNA and your RNA with the various differences. Now, DNA and RNA. DNA, we're going to look at DNA first, and we'll come back to RNA a bit later. Now, DNA stands for deoxyribose nucleic acid. Deoxy meaning that it has a lack of oxygen. Ribose meaning that it's a sugar that has a lack of oxygen. Nucleic acid means that it is an organic molecule, and this organic molecule will also play a role in protein synthesis. So it is sort of just a whole bunch of connections that you must form. It is a deoxygenated sugar, right? In a nucleic acid structure where DNA is an organic molecule and also plays a role in protein synthesis. Subsequently, RNA is not deoxygenated. It is a ribose sugar. It is a nucleic acid. When it's a nucleic acid, ultimately it means that it's an organic molecule, and it also controls or plays a role in protein synthesis. So those are the various deductions that you need to make throughout this video. Now, the location of DNA. DNA is found in 4 places. I think there is more, I'm not sure. I think there's a 5th place, but for grade 12 knowledge, DNA is located in 5 places. DNA obviously is found in the nucleus, which we discussed. It makes up genes on chromosomes, right? And we discussed that where you have the chromosome, you have the part of the DNA, and then the part of the DNA segment is known as a gene. And we'll look at it also in depth a bit later. It's also found in the mitochondria, where it is known as mitochondrial DNA. And mitochondrial DNA will play an important role in evolution. It's another topic, we do this here a little bit later. But mitochondrial DNA will also play an important role in evolution. And also we find DNA in the chloroplast of plants. DNA, and you might say, but isn't DNA only for humans? No, it's not. DNA is found in every living organism. And we're going to explain, and sometimes the argument will come that if DNA is found in every living organism, why don't we all look the same? And we're going to look at why that is why not looking the same is the case. And also DNA that is found outside the nucleus is known as extranuclear DNA. Extra meaning outside, nuclear meaning outside of the nucleus. Right, now this is just a note that I want to bring across here. Chromosomes, which we discussed, which looks like this, depending, this is replicated, so this would have undergone DNA replication, right? Along thin thread-like structures, they contain DNA. They are wrapped around histone proteins, and histone proteins are there to provide shape to the DNA and to protect the DNA from being broken or lost, right? That is just some information. But what I want to look at is I want to look at this. So this here is the nucleus, right? We've discussed that. From the nucleus, we get what is called the chromosome. Now, if you take one of these chromatids and you zoom in to it and you unwind it, you will be able to start to detect this DNA structure. The DNA structure, and this is how we get the DNA structure. So it will unwind. Remember here, the DNA is severely condensed, right? The DNA is piled on top of each other like that. At this point here, the DNA is piled on top of one another like that. Now it is going to unwind. It's going to unwind significantly. And with that unwinding, here we see the histone, right? Now we can notice here that the DNA is wrapped around that histone. If you just look at that thread-like structure, it is wrapped around the histone. It's wrapping around it like that, right? And then from there you get DNA. Now can you see they have made it, they have showed us there's your DNA and there's your DNA. But remember this whole segment here, this entire segment here, and continuing infinitively is DNA. But a segment, this is the important part, a segment of the long DNA chain is known as a gene. A segment, grade 12 need to remember the difference between the gene and the chromosome, the DNA and the gene. The chromosome, if I explain it again, the chromosome has DNA all stacked up. If you think of when we make a fan, when you make a fan with a paper, when you fold it, you make all those folds. That is DNA stacked on top of each other. When you open the paper, say you want to draw on it now, it's not a fan anymore. When you open the paper, that is now your DNA, your infinite DNA. And so you folded it on top of each other. Now you're opening the page, and now it is now has various segments. This entire thing is DNA, but each segment, grade 12s, is a gene. Each segment, dear grade 12s, is a gene. Please keep that in mind. If they ask you in the exam to differentiate between the DNA, between the chromosome and the gene, you need to be able to understand what they are asking you, right? And what is the severe differences between it. Now I'm going to go into a gene. You can see that I've made an extra note on the gene, and I'm going to go into that a little bit later. And then another important structure. So you can see that we are zooming into the structure more and more and more, right? We went from the nucleus to the chromosome to the DNA which is called the nucleosome. We went from the nucleosome and we went and zoomed in further, and we got a gene. And now when we zoom in on the gene or on that segment of DNA, what we will notice is that there is a structure called a nucleotide. And I'm not going to speak too much about it because I will get into it a little bit later. But the nucleotide is the smallest structure in this whole topic. It is the structure that makes up the structures. So, but there is a segment, I'll explain it in detail. Going back to the gene, a gene is short segments of DNA, which I explained into great detail just now. It's short segments of DNA. What a gene is, why genes are important is because genes code for specific proteins. Now, our body is made up of proteins. Our hair is proteins, our nails are proteins, our hormones, some hormones are proteins like insulin. Insulin is a protein consisting of 51 plus amino acids, and we're going to look at that a little bit later. But there are various structures in our body that are proteins, and that is why DNA is important, because DNA needs to give the code that will start making these various proteins. But it codes for particular proteins, it controls inherited characteristics. Ever wonder why you look like your mother? Ever wonder why you look like your father? Ever wonder why your grandmother has diabetes, and now your mom has diabetes, or your dad has diabetes, and you will probably have diabetes? Is because of the inherited characteristics that are passed down from gene to gene, right, or from generation to generation. You will have the starting generation, and that generation would have passed off various things, and it will go on like that forever and ever and ever, unless this will be a side note, but where does this change? How can we change it? It is something called the human genome, and I'm going to discuss it a little bit later, or genomic medicine, as we now like to call it, or genetics as well. But the important part here is that with the advancements in artificial intelligence, with all the advancements recently, it is possible, if you have the money, it is possible for you to go to a genomic clinic, have them zoom in on your genetic code and have them alter it. But there's obviously risks with that as well. And if you are religious, that brings in the argument that aren't you playing God? Because the basis of religion, dear grade 12s, and this is an important concept because you will deal with it extensively in evolution. The basic concept of religion is that God gave you what you need to work with. It is not your responsibility to change it. Anyway, and then the last thing here, it determines the organism's appearance and functioning, which are highlighted on. If you look like your father or your mother, a lot like your mother or a lot like your father, it is because the genetic information that came from your mother or your father is extremely strong. Like I have a lot of genetic information from my mother, so I look more like my mother, or I look more like my family side, my mother's side of the family, right? And that is your gene. So if they ask you to define it, you will say a gene is a short segment of DNA that codes for particular proteins and controls inherited characteristics, thereby determining organisms' appearance and functioning. That is the function of DNA. If you had to define the function of a chromosome, you would say it is a long, thin, thread-like structure that contains DNA and is wrapped around a histone protein. If you have to describe DNA, you will say that DNA is a type of nucleic acid that consists of a deoxygenated sugar, ribose sugar, or deoxyribose. Right? That is the various differences, dear grade 12. Now, let's quickly go on to the discovery of DNA. Now, who discovered DNA? There were three. Okay, in this one, there were three, but recently it now became four. It was always four, but they are now accepting. So I'm just going to add here as well. Right? Now, we need to understand the differences here. If they ask you the question. Now, I'm not going to go into all of it, but I just want to know the question. If we look at, if the question asks, who discovered the double helix DNA structure? That structure that looks like this. The double helix DNA structure, you can put down all four names. Grade 12s, please take note. If they ask who invented the double helix DNA molecule, who invented the DNA structure, who constructed the DNA structure, who constructed the DNA double helix, They can ask it in various ways and use various synonyms. But the point here is that if they ask about the structure, who discovered its structure, who made up the model, you are allowed to say all four depending on how much they ask you. If they ask you for three, you can write any three. If they ask you for four, obviously you'll have to write all four. However, if they ask you who took the X-ray of DNA, this is where the difference comes in. If they ask you who took the X-ray of DNA, you are only allowed to say Rosalind Franklin or Maurice Wilkins, because the story goes that Rosalind Franklin and Maurice Wilkins, they discovered the X-ray of DNA. When they discovered the X-ray of DNA, Maurice Wilkins decided to give it to Francis Crick, and then James Watson and Francis Crick decided to make the double helix molecule, the double helix structure of the molecule. Right, that's how the story goes. Ultimately, they are only supposed to accept James Watson and Francis Crick, but because of 2024 national senior certificate examinations, they are now accepting all four when they ask for who discovered the structure, and only two when they ask who took the X-ray. So please be able to differentiate in your question between the two. Now we're going to get to, this is my favorite part. It is the structure of DNA. Now what I want us to note here is that DNA is the large unit. That is what I want us to note here, that DNA is the large unit, right? The DNA molecule, which we affectionately call a polymer, right? Which I noted here. It is the polymer. Now, what we know about polymer, about a large molecule, is they need to be made up of smaller units. And these smaller units are called nucleotides, right? So, ultimately, what we can say is that nucleotides are the building blocks of nucleic acids, right? Or the building blocks or the monomers of nucleic acids. Now, DNA, dear 12s, or nucleotides, dear 12s, has two categories, depending on what you are explaining. And we're going to look at that in depth. Whether you are explaining DNA replication or protein synthesis, there are words you need to use. You're either going to say DNA nucleotides or RNA nucleotides, right? So you will have DNA nucleotides. I'm just going to write the letters, which we'll look at a little bit later. Or you're going to have RNA nucleotides, which I'll get into later, right? And depending on which topic or what process you are explaining, you will need to either say DNA nucleotides or RNA nucleotides, and we'll look at that a bit later. But simply put, if they give you in the biological terms, they like to give this in biological terms. They're never going to ask you for the definition. This is a common biological term question, right? Where they will just say the building blocks of nucleic acids, and obviously you will write nucleotides, or they will ask you the building blocks of DNA, you will say DNA nucleotides. The building blocks of RNA, you will say RNA nucleotides. But they like to give this in biological terms. Please take note, your grade 12s. Now, we just want to quickly look at so I remember I said this is the smallest unit. Now we're going to zoom in on that unit. Now that unit is made up of three components: a phosphate group, a sugar group, and nitrogenous bases. Right? This is the DNA. So this is a DNA nucleotide. Right? RNA nucleotide doesn't look different, but there are certain things that change. Now, in a DNA nucleotide, remember that the sugar is known as deoxyribose, whereas in RNA it is a ribose. And then also we need to note here that we have nitrogenous bases, ATGC. Right? ATGC. Now, these nitrogenous bases are important because these nitrogenous bases is your genetic code. Your code, I don't know, obviously, I don't know how our codes run, but your code will run with these four letters continuously. It will run like that. And all of these letters, dear grade 12s, that all of those letters combined, those are the letters that are determining the way you look, the way you talk, the way you everything. The whether you will have an abundance of a certain hormone, whether you will have a genetic disorder, whether you will have a genetic illness, which is there's a difference, right, between a genetic disorder and genetic illness. I'm not going to get into that, that's not important, but there are differences. That is the code. That is why the topic is called the code of life or the genetic code, right? And that is your nitrogenous bases. Now, DNA has 4 nitrogenous bases. Now, these nitrogenous bases is where adenine is one of it. Now, what we need to notice is that adenine will always bond with thymine. A always bonds with T. G always bonds with C. Guanine always bonds with cytosine. And what we want to note is that adenine and guanine are known as purines because they are large molecules. Thymine and cytosine are known as pyrimidines because they are smaller molecules, right? Because they are smaller molecules. And as I read here, note that adenine always bonds with thymine and guanine always bonds with cytosine. Important to note, in DNA. In RNA, there is something changes, right? In DNA, adenine bonds to thymine, guanine bonds to cytosine. Do not get it confused, right? Because in RNA, something different happens. So something is added, something is taken away. And what is important to also note is that they are bonded by weak hydrogen bonds, which are these bonds over here. These weak hydrogen bonds. And the reason why these hydrogen bonds are weak is so that enzymes can easily break them during DNA replication and transcription. Transcription is the first part of protein synthesis, right? So that you can just say that that is part 1 of protein synthesis, dear grade 12. Now, 2 bases together are called a base pair, right? So A and T together are called a base pair, but because they always bond with each other, we say it is a complementary base pair, right? What do we mean? What does it mean when we say it's a complementary base pair? All we are saying, the grade 12s, is that it can only bond with that specific nitrogenous base. They complement each other. G and C complement each other. A and T complement each other. A and G don't complement each other. And T and C don't complement each other. So please note that this is known as a base pair. However, if it is bonding to its proper molecule or proper nitrogenous base, it is called a complementary. A complementary base pair, right? So, also another thing that we want to note here is that 2 bonds form between A and T, which we can see over here. You can see it's only 2 nitrogenous bonds. And 3 bonds form between G and C, which we can see there. And this lends itself, or should lend to the knowledge, dear grade 12s, that if A and T has 2 bonds, and G and C has 3 bonds, it means that it's going to take longer for the enzymes to break the bond between G and C, whereas the amount of time it will take to break the A and T hydrogen bonding. Right? So, now, as we said here, in a DNA molecule, the number of adenine will always equal the number of thymine. Likewise, the number of guanine will always equal the number of cytosine because they are complementary. So if there is 20% adenine in a DNA molecule, that means there is 20% thymine, which means together that is 40%. 40% of 100 is 60%, which means that if you divide the 60% by 2, guanine, cytosine, or let's say, is going to have 30%, and guanine is also going to have 30%. I hope you follow. Right? Adenine has 20%, therefore thymine has 20% because they're complementary. Right? The amount of adenine, if there's 4 adenine in DNA, obviously there's going to be 4 thymine as well. Right? Now cytosine and so that means it is 40%. Adenine and thymine make 40% of the DNA molecule. 100% minus 40% is 60% degree 12s. Therefore, if you divide the 60% by 2, cytosine is going to have 30% and guanine is also going to have 30%. I hope you follow that, dear grade 12s. Right? I hope you follow that, dear grade 12s. Now, what I also want us to note is there's something called the sequence of base pairs. The sequence of base pairs. What is a sequence? Dear grade 12s, we have been doing sequences in mathematics. Pure maths. A sequence means that it is running in a certain order. Dear grade 12s, there is order to it, there is structure to it. So if we look at the sequence of base pairs for this image at the top, let me just zoom out a bit. If we look at the sequence of base pairs for this image from the top, if we're going from the top here to the bottom, dear grade 12s, oh, this makes me excited. To the bottom, dear grade 12s, can we note it's A, T, G, C, T, A, C, G. That is the sequence of base pairs. Right? That is how the nitrogenous bases are running from top to bottom. Dear grade 12s. Now, the sequence of base pairs is important because it provides the code that gives instructions for protein synthesis. And this code, which we have discussed already, is important because it's your genetic code. Remember, I said that it will sort of run like that. Right? Now, this here is a code. And what this code does is this code tells the body to make a certain amino acid. Right? I'm getting a little far now. To make a certain amino acid. Or amino acids. Right? We're going to look at why I'm saying amino acids a bit later. Certain amino acids. Right? And those amino acids will then join and form a protein. But that's not important right now. That is not very important right now. Let's get back. But why? If they ask in the exam, discuss the importance or why is the sequence of base pairs important, you will say it is important because it provides the code that gives instructions for protein synthesis. That is all you need to know. And we're going to look at why. You know, in the beginning, it all seems all over the place. But when you come to protein synthesis and replication, you will understand how it all blends into one another. Now, this also, I just want to go on the concept of why we say it complements one another. Right? Complementarity, or whatever the English word is. Right? Gelykendheid, sal mense soos sê in Afrikaans. Anyway, so if in the exam they give you strand A, which is the strand over here, and they ask you to write the complement of it and label it strand B. Obviously, if it's a DNA molecule, you will note that A will bond to T, G will bond to C, T will bond to A, and C will bond to G. Therefore, strand B will be the strand here. And that is why we say that the one strand complements the other strand. What do we say when we say it complements? It means that it doesn't match. We can't say it matches because then it would be AA. But what we are trying to say is that it's pair. If I say two people complement each other, it means that they fit together. Right? They work together. Thereby saying that if I say A is complements T, it means that they work together, that they are bonded together. They must always be bonded together, dear grade 12. And therefore we can deduce the statement that strand A complements strand B and strand B complements strand A. Grade 12s, please go and look over non-coding DNA in your textbook on page 1.5 on part 2, because non-coding DNA blends into what is known as DNA profiling. Please, it's not called DNA fingerprinting. Some textbooks might say that that's an old understanding. It's DNA profiling. Right. Go to the next video where I will discuss DNA replication in depth.